Thank you, Martin. Good morning and welcome, everyone. This plenary aims to address advanced or emerging public health surveillance tools and techniques using practical and recent examples. We will be discussing 3 exciting topics with our distinguished speakers. First, the current uses of whole genome sequencing in surveillance and outbreak investigations. Then signal detection algorithms applied in surveillance and adjusted for post-pandemic changes. And finally, recent advances in the use of artificial intelligence in surveillance and prevention of healthcare-associated infections.
And to cover these topics, we count with the presence of our speakers, Dr. Lieke van Alphen, expert in molecular epidemiology. She is the head of the Regional Typing Laboratory in Maastricht University Medical Center, and her research focuses on antimicrobial resistance and next-generation sequencing for rapid data sharing. We also have Dr. Andre Charlett. He is the head of the Statistics, Modeling and Economics Division at UK Health Security Agency, where he and his team develop methodologies to contribute to preparedness and response, and is part of the Scientific Pandemic Influence Group on Modeling. And Professor Caterina Rizzo, a public health expert and professor of hygiene and preventive medicine at the University of Pisa, with research on infectious disease epidemiology, preventing and risk communication, and part of the Italy's National Immunization Technical Advisory Board. So this is our panel, and we also want to remind you, Martin has just said it, that you can help us with this session and contribute with your questions at any time using the app. Go to your app, just know that there is the comments and the questions and answers, and even if we will be keeping the time, your questions will be read and some of them will be fed in the conversation and sent to the speakers.
And now it's my pleasure to invite Lieke to talk about the latest developments in the field of whole genome sequencing, including the improved identification of infection clusters and management of outbreaks.
Thank you very much for the kind introduction. I'm working at a university medical center, so really at a regional level. So I'm really focusing as well on the challenges that we encounter doing whole genome sequencing in this regional level. So I really want to take you along sort of in the whole adventure of whole genome sequencing at a national or at a regional level and in a local hospital. So of course, everybody here knows whole genome sequencing is really about analyzing the whole genome of a microorganism. And doing this in an outbreak setting, it really leads to earlier detection and more precise investigation of outbreaks. And this combination of the whole genome sequencing data with the epidemiological context really gives us a strong advantage in doing outbreak investigations. Of course, we can also characterize the microbes a bit more and really look into their ecology and transmission. And we want to identify unknown transmission routes that epidemiologically might not have come up in a hospital, but that we can then still identify using whole genome sequencing. So of course, like when you want to do sequencing, like what do I need? Of course, you really want high accuracy and good discriminatory power, but you also want a low price and a rapid turnaround time. And I think, as we all know, if you want something good, you want something fast, but you also want something cheap, that's typically quite difficult. Like if you get 2 out of 3, you're typically already quite happy. So I'm just trying to explain today that hopefully we can get there to actually have good data for a cheap price in a really rapid turnaround time. If you look at bacterial typing methods that have been used for outbreak investigations, they've really changed over time, and there's a lot of different methods that you can use, and they all have a different discriminatory power, so they really have a different resolution that you can use. And I think the most recent ones, of course, are whole genome sequencing, but before that, pulse field gel electrophoresis was used a lot because that's a relatively cheap method to implement, and it did give quite a high resolution. If you then want to look into how whole genome sequencing actually enhances these results. If you just have a whole unsorted population of different pathogens, and some of them are related, and you want to sort those, if you do pulse field gel electrophoresis, you can really nicely sort those into big groups, but you will also include isolates that are not actually part of your outbreak. So that, of course, becomes a problem if you really want to look in transmission. If you now use whole genome sequencing, you can more accurately actually make those groups, so it will also be more easy to identify the true clusters and to really look at where your transmission is happening and how it's going on. Just as a reminder, I think all of you know this, if you do look at the whole genome sequencing workflow in the lab, there's a lot that comes into play with this. You really have your lab routine where you isolate your DNA, and then you have your whole genome sequencing method where you can choose your methodology, the long read, short read, different types of platforms are available, and that's all like what you want to do and how you want to do it. Then, of course, when you have your data, you have this data processing and analysis where you really need to do bioinformatic analysis on your data. And I think, as we all know, it's really important to get good data, because as soon as you start doing analysis, like garbage in, garbage out, as we all know. But then the data processing itself, also there's a lot of different tools that are available which are constantly in development, with like new base colors, new tools for doing your genome assembly, for example. And I think there's also a lot of time and a lot of accuracy to be gained. And of course, with the data processing, we can nicely look into clusters, but we can also look at antimicrobial resistance genes and virulence genes. But then the reporting is essential as well. Your data needs to go where it's actually needed and where you can use it for action. And I think also one of the difficulties, especially in the local lab or in a regional level, is that you need all these different specialties, preferably in-house. So you need, of course, your wet lab, which typically is quite well organized. Then your data analysis, which is not always available in the lab. To have your own specialist for data analysis that can really sort of cater to your needs that you want to do in your hospital. And then with the reporting as well, when you want to report to your clinicians, it also needs to be like reporting that they can understand and take the proper action for. So there's a lot of different specialists involved, which sometimes is also one of the hiccups, I think. I want to give a couple of examples that we've been doing in the recent years, just to show like how we sort of developed it and what we're doing currently in the regional level. And I think for us, the high throughput sequencing really took off with COVID. As I think for a lot of different labs, like that's when you all of a sudden started doing loads and loads of sequencing. And this also showed really nice opportunity to give a good picture of regional surveillance. So you have your isolates and you really show like which variant is coming up. Is there a new variant coming in my region? And what do I do with this? And if we had this regional context, it also made it easier for us to really look into our hospital outbreak investigations, where you look in a lot more detail, like which of my patients actually have exactly the same isolate that only differ maybe 1 or 2 mutations. Like, is there spread going on in my hospital, or is it just all the patients that we just have admitted to the hospital? So, for example, in our hospital, we had like different clusters of different genotypes all in the period of the Delta variant of COVID. And this was actually during a strong period of lockdown in the Netherlands. So there was not much virus circulating anymore. We actually saw decreasing numbers of infections. And then they decided to actually start making sure, they wanted to release the lockdown so people could go out again to bars and restaurants, because they thought this was time to now really release those measures. And they also had like a vaccine campaign where they said, like, if you get full vaccinated, you can get your QR code so you can go to restaurants. And they had this for the Janssen vaccine where you only needed 1 vaccination. So what we then saw is that you see here in the pictures on the right, there was like really a drop in the number of positive tests. But then after actually they lifted the measures, people started going out to bars and restaurants again. And after that, we saw a really strong influx of certain genotypes in our regional surveillance, but also first in our hospital. So together with our municipal health services, we really went into an investigation of this in different bars and restaurants. Like on the picture on the left, you see all different venues. And we looked at the different genotypes, like where people had been, how people had moved from one restaurant to a bar, and really showing like where people had been. So the people admitted to our hospital were interviewed for this, like, have you been to a bar, have you been to a restaurant? And we could really see that some genotypes were really closely related to one specific bar, and other genotypes were really more like across bars and really transferred. And then a couple of weeks later, we saw this also popping up in our regional surveillance. So we saw these genotypes that made like up 40% of our hospital population right after this lifting of the measures. We also saw that popping up in our regional surveillance. So that was really interesting for us, sort of to have the correct context of your regional surveillance and then also link that to your hospital outbreaks. So this is what we also wanted to do for our bacterial, like hospital outbreak investigations, to make sure we had a good regional context so we can actually say something about our hospital outbreaks. So then we were really looking into, like, can we do this data sharing? For COVID, it worked really well. With like a next strain, GIS8, like you could share your data, you could really see your context. But for especially for hospital pathogens, it's not as easy. Because national surveillance is being done, for example, for the carbapenemase-resistant bacteria, and for MRSA in the Netherlands, very strong surveillance. But all the ESBL bacteria that we also worry about in our hospital, there's no like sort of national surveillance. So we wanted to see whether data sharing would be a good option and that we could just share our whole genome sequencing data from the bacteria. So at first we did one experiment, we'd already done that with the harmonization of the lab methods and then using the same analysis tool, seeing if you could share your data easily that way. And when we did this, we saw if we did the whole genome sequencing in 3 separate labs, and we used the same analysis tool, if you do a whole genome MLST analysis, you only saw like 1 or 2 alleles difference typically. So it's very easy to do the sequencing in different labs, and then if you combine your data analysis, this works well. What we did see is that if you did de novo, if you use different de novo assemblers for your data, you did introduce a lot of variety, up to 150 SNPs, and there you might actually be surpassing your threshold of outbreak investigation. So that was quite worrisome. So therefore we wanted to see if everybody uses different tools, can we still share the data? So then the special interest group for bioinformatics in the Netherlands did like a big investigation with all the labs that were typing for these bacterial ESBLs. And they really wanted to see if we send everybody the same set of data and say, like, pretend that this is an outbreak in one ward, or this is the data you get from one ward in your hospital, like, is there clusters going on or not? So that's what we did. And if I show the picture here for the VRE, we did this for VRE Klebsiella. The picture, each black dot is an isolate, and then there's links between the dots. If there's like a green line, then all the centers agreed that those 2 isolates were linked. If there is a blue line, then the majority of the centers actually agreed that those 2 isolates were linked. And if you see a red line, then there was discrepancy. There was not a majority vote that this would be a linked isolate. And what you can see is that there was a lot of difference in interpretation. Like, there were not many isolates where people actually agreed, like, yes, this is a real cluster. And this was even when people used almost a similar bioinformatic analysis tool, they still saw quite a bit of big differences in clustering. And we also saw there that if you did stringent filtering, some centers also failed to detect some of the AMR genes, for example. So this made us worrying, like, okay, we really want to share data in our region to have a good context for surveillance and to really link our hospital outbreaks to our surveillance. But we really need to make sure we use the same tool. So in our region, we then set up a regional surveillance system where we have an online tool where we can just upload our raw data and then really do the analysis in this tool. And we're now doing this rapid regional data sharing for infection prevention in our region. And one of the interesting things, of course, that come out, you can see either clusters within hospitals or possibly also between hospitals, but you also see influx of new variants of a species you're looking at. In the Netherlands, we do hospital surveillance for vancomycin-resistant enterococci, and recently we saw that a novel sequence type was detected, sequence type 1299, which is in Germany, it's more endemic, and it does cause nosocomial outbreaks. So we saw this in our hospital and actually caused an outbreak in our hospital. And we had quite a few isolates that were very strongly related, as you can see here, with only 1 or 2 alleles different. And then one of our neighboring hospitals that sends in isolates to us for typing, they also saw this same isolate. So of course we were like, oh, do we really have something that's spreading to hospitals in our region? But because we shared this data this rapidly, we could see it's a completely different type and it was not related based on the core genome MLST. And I think here it's really important, like now we're comparing multiple different organisms in this regional setting, and we really have to keep in mind, like you have different genomic thresholds for different organisms, different species have a different mutation rate, different contexts, and some might be hypermutators. You also need your sort of regional context if you see all of a sudden an influx of bacteria again. For example, we had an outbreak with VRE ST203. We had an outbreak for a while, it was completely gone for half a year to a year, and then it started popping up again. So we were worried either it's still in the environment of the hospital or we have a new influx of this bacterium. This was the early days for us for whole genome sequencing, so we didn't have a lot of regional data, we didn't have a lot of context, so we asked people from the Danish Institute, the Serum Institute, to really compare it with their data set. So they put our Maastricht samples in their context and they really saw that those isolates that they had either from Danish origin or from German origin were closely related. So this is just a clone that's very successful in going around. So it's not something that was just specific for our hospital. And then it's also really hard to say, is this an outbreak in your hospital or is it just a successful clone that's being imported at several times? So just some last words on the sequencing technique considerations. I think we all know, I've talked now about clonal spread of bacteria, but of course you can also have plasmid spread, as we've heard presentations about yesterday as well. But the plasmid is actually moving between bacterial isolates and also possibly between patients. And with the short read data, you can't really see those well, because if you do the data assembly, you don't get really your data nicely assembled into a genome and separate plasmids. So we always used to do hybrid assembly for this, using long read and short read data, so you can nicely see your genome and your separate plasmids. But now, more recently, the nanopore sequencing has become a bit more advanced and the quality has become better. So now you can just use nanopore-only data to actually see these genomes. And that makes it easier to actually do this analysis, and also a lot cheaper and faster. So I think this is really nice to see this. I'll finish with a last example that I just want to show. It's a recent example we have from our hospital, just for the case of how to showing a plasmid spread. We had a patient in our intensive care unit that actually already arrived in the hospital with a carbapenemase-resistant Citrobacter, and this actually turned out to have an NDM7 gene. And this patient was quite long admitted to our hospital, and over time, actually in clinical cultures and in surveillance cultures, we saw different isolates popping up that also carried this NDM7. And over a period of like 158 days, we saw 4 different organisms in this patient, all carrying the same plasmid with this NDM7 gene. So this is really showing that this plasmid is very successful actually moving between species. And then later on in our hospital, we did a point prevalence screening where we screened all the patients on sort of in 1 week for ESBLs and for carbapenemase-resistant bacteria. And there we also saw extra patients popping up that we hadn't before seen, also carrying bacteria with the same NDM7 gene. So we did whole genome sequencing on all the patients we found, because of course now we were really worried that we might be spreading this plasmid in our hospital. We did see some other cases, but most of those were import cases coming from abroad, as was our first case as well. We did see, however, when we did environmental sampling in our intensive care unit, that we also saw in the environment of our intensive care unit different species popping up. And the color scheme here actually shows which isolates were clonal and which isolates were not related. So we had Citrobacter, Klebsiella, Enterobacter, all with the same plasmid, which is now also in our environmental sampling in the intensive care unit. So luckily, since August, we haven't seen any new patients anymore, and we have our new prevalence screening coming up. So I hope this was just sort of a one-time thing, and that is also slowly now moving out of our environment in the intensive care. But for us, this is really important because even though like national surveillance is being done for carbapenemase-resistant bacteria, environmental sampling samples you cannot sort of send to the reference center. They only do patient samples. So for us, it's really important to also have our own context and to also be able to do this sequencing in our own institute. So just briefly, my conclusions, I think there's a broad set of expertise that you need for whole genome sequencing, and you really need your surveillance data to provide context for your outbreak investigations. And data sharing is really essential, but you really need to consider how you're doing it and how it's working. So thank you. That was what I wanted to share.
Thank you very much, Lieke, for this excellent talk. I have 2 questions to you. In the interest of time, I will give you both questions now. So first, how can public health institutes with limited budgets and limited resources extend or adopt the use of whole genome sequencing? And the second point I would like you to address is how can well-interpreted whole genome sequencing data help us to make better decisions? Can you please provide some other examples?
Yes, yeah, I think for your first question, to actually to implement this whole genome sequencing, I did bring a slide on barriers to implementation. If they could show that on the screen, I think, yes. So there are, of course, a lot of barriers to implementation to implement whole genome sequencing in a certain setting. And I think a lot of the national public health institutes are already doing this, and they also have the funding. But typically lack of funding, but also gaps in expertise are really important to actually bring this to the lab and to implement it in a meaningful way. Like, for example, bioinformatical knowledge is really necessary to be implementing this in a proper way. And that's not always available. And of course, data dissemination, sort of feeding it back to the people that actually need it, is really important. So therefore, I think training is essential. And also what's been identified as one of the needs would be an all-in software where people can sort of bring their data together and have like a similar analysis, so you know you're actually looking at the same thing. This could also help with like cluster definitions, for example, and how to call data. And I think there has been really nice initiatives, of course, with the European Food Safety Authority and ECDC. They are sharing data now for the food safety, and I think that is really working well. But there's also not all the countries are doing as much sequencing yet. So there you also see differences, that some countries are contributing a lot of data, while from other countries there's only few data available. And that also skews, I think, the context a little bit, if the data sources are just coming from a few countries that are sequencing a lot. So I think there we could achieve a lot more. And I think there, of course, capacity building is really essential. Like really making sure you get the expertise at the level where it's needed. And I think like with the HERA project for SARS-CoV-2 and also now for other pathogens, there's a lot of capacity building within Europe going on, and a lot of training of people and also bringing the expertise where it's needed. And to address your second question, there was more on the implementation and sort of the how to get the data for action at the right site. And I think for me, it's all about also doing your rapid data analysis, having it when you need it, where you need it. I'm a big advocate for doing like regional typing and then sort of sharing that with the national level, because you prevent sort of sending back and forth clones. And nowadays with the data, it is easy to share the data rapidly. And you can also include other data that might not always be involved in a national surveillance system, but might be useful for national surveillance.
Thank you. Martin, do we have any questions from the audience? Would be nice to see what are the most pertinent questions to Lieke.
Well, we actually have 2 questions. And the first one, if maybe you could be very specific.
Oh.
Can this tool be used for monitoring or surveillance? Or an and, I guess. How quickly can sequencing technology be utilized? And how much does it cost? Very concretely, if you wouldn't mind. What advantages does it offer from a surveillance perspective? Okay. So I think that's 4 questions.
Yes,
but it's all wrapped
4
up
questions.
into one. I'll give you the tablet so you can see it.
Yeah, I think like using a sort of a rapid data sharing tool, you can definitely use that for monitoring or surveillance. If you share the data quickly and people are trained to sort of go online and really look at this data and use it. But of course, for us at a regional level, like we're not sharing a lot of metadata with it. We're not sharing a lot of epidemiological data. So what we're doing, we identify clusters, and then of course, if we see possible clusters, we start talking to the infection prevention team of the different hospitals to really start talking about which patients are these, where have they been, and they start really doing their epidemiological investigation as well. And I think in general, like how quickly you can set up sequencing, for example, if that's what's meant with this question. Like it's nowadays, especially with nanopore sequencing, it's relatively easy to set it up. You don't need very expensive equipment. You don't need that many different things in your lab. And also there's a lot of data analysis tools out there that are free that you can just sort of play around with and start using. So it's relatively easy and not very expensive, I think, to set it up.
Okay, so not very expensive.
Well,
could you quantify that?
Yeah, it really depends, I think, which method you use, what you use it for, right, and also how much in bulk you're doing your things. As soon as you start sequencing in bulk, of course, it becomes a lot less expensive, because then you can do a lot of sequencing at the same time. So if you want to do like small hospital outbreaks of like 12 isolates at a time using short read data, you might be looking at €150. If you're doing that with nanopore sequencing in smaller settings, it might go down to like 50, €75 per isolate. And I think it will decrease further in costs as well as we progress in time and methodologies are changing.
All right, then very quickly, and I'm going to ask you to read this with me, just to make sure we get it right, from Florian, who you might know. Plasmids. With WGS short read problems, do you go back full circle to phenotypic AMR testing and look for the combination of resistances? Is that clear, the question?
Not completely, to be honest.
Is Florian in the room or are you online? If you're in the room, put your hand up.
I think he is here, yes.
Florian, we're going to very quickly go to that mic over there. It's just over here, and then you
It's
can.
an interesting question. I'm just not sure I'm understanding it correctly.
Yeah. Yes, it's a little bit of a. It's a bit like trying to get through the snow, isn't it?
Thank you. I mean, antimicrobial testing used to be done on the agar plate, and then you have one isolate, and then you look at the combination of resistances. And when you have a particularly characteristic plasmid which carries a very specific set of resistances, and there would be 2 ways: either looking at it phenotypically, like looking testing actually the resistance, and the other thing is looking for the genes. And if you say that assembling the genes is getting difficult with certain technologies, then So you don't know whether it is in the core genome or whether it's in the plasmid. Oh wait, that's a stupid question. You still don't know whether it's in the core or in the plasmid. Okay, but wouldn't this stress a more important role of testing than the resistance patterns phenotypically and compare this, like old school microbiology, wet microbiology?
I think that's definitely, we always need to remain, or at least we should be testing phenotypically currently as is, because not for all the genes it's known exactly like how much resistance they cause. It's not always as clear for the genes as well if they just have slight point mutation, how functional they are, how well they're actually doing that. So I think for functionality testing when genes are first discovered, definitely we need phenotypical testing. As soon as sort of a plasmid has been really well described and well established, and you've shown that if you put this plasmid in a new bacterium, it really shows this phenotypic profile that you expect, then after that I think you can just use the data for seeing if it actually transfers to other bacteria. But also in a different bacterial background, it might show different levels of resistance, for example. So yes, definitely, we're not getting rid of phenotypic testing yet.
Thank you very much. Thank you for the question as well, and for moving so quickly to the microphone. Much appreciated. We've got a lot more questions coming in, but I think let's move on, and then if we've got some time, we can take them at the end.
Yeah, thank you. Thank you. Yeah, in the interest of time, we can move on. Thank you very much, Lieke, for your presentation and your answers. And now the floor is yours, André, to talk about advance in algorithms for signal detection.
All right, good morning. So I'm going to go through using an example, really, of finding outbreaks using surveillance data. The example I'm going to use is what we do in UKHSA and what we've done for a while in UKHSA, but also trying to understand shocks. And the obvious shock that I'm going to go through is the COVID pandemic and the impact of lockdowns and other regulations that were in place during that pandemic. So there's about 4 broad areas of work here. The first 2 sort of papers there, they're concerned with methodology development. So one area that we have worked in, the first paper there goes back nearly 30 years now. I think 3 of them have retired on that paper. I know Mike Catchpole's just retired. Paddy Farrington retired a few years ago. Nick Andrews is still not retired. He's younger than me, actually. So that's where the start of what we were doing back in in the mid-1990s in the then Public Health Laboratory Service. We got asked the question, can we detect unusual patterns of reporting that could indicate the presence of an outbreak occurring in the population? So with that motivation, they set up what was the original Farrington algorithm, and that's been adapted over time. The second activity is reviewing the methodologies that have been used across different public health institutes across the globe. That's really quite an important thing to do, to understand what methods have been used, what their performance was, etc., what they were trying to do in those papers. So it gives you an idea of all the plethora of methods that are available to you. Another activity is evaluation of performance. All of these methods will have, they'll all have different performance characteristics. And it depends on what you want to do. It's the same in sometimes you want to maximize the sensitivity, maximize the specificity, improve the timeliness of detection. So you can look at sort of these types of papers where people are reporting how good each of these methods are, or a range of methods are. And finally, and probably one of the most important things, is making that code available. So packaging up code, putting it on public repositories that anybody can pull down and use. So the last paper there was the RKI initiatives on the R surveillance package. So a lot of the techniques, a lot of these methods are available through that software. I'm going to move. So this is one of the techniques. This is the technique that we developed in the early 2010s, what's come to be known as Farrington Flexible or Flexible Farrington. I'm going to say I've got no equations in these. There's no statistical equations here. So I'm going to go through this and just trying to explain what this algorithm does. It's a fairly simple algorithm, to be frank about it. It's not that complicated. Most of you have been through things like multivariable analysis modules in your EPIET training. These are the sorts of things you'll get taught. This is a quasi-Poisson model. Why is it a quasi-Poisson model, not a Poisson model? Because invariably, every time series from infectious disease surveillance has more variation in it than you'd expect from purely from a Poisson process. So we fit these quasi-Poisson models. It's only got essentially two components, a trend, a temporal trend, and a 10-category factor in there to replicate or try and model the cyclical patterns that you see. So it's flexible. It can cope with a whole variety of time series. And these are applied to several thousand time series every week in UKHSA. So one of the things we try and do here is maximize the specificity. We don't want too many false positive detections. The epidemiologists don't like to wade through a whole long list of flags to say this looks unusual, this looks unusual, this looks unusual. So we try and minimize what the false positive rate is in these algorithms. Epidemiology is really driven, or a lot of it, for most pathogens, is driven by how populations mix. So you can see here on the left-hand panel, this goes back again into the early 20s. These are polymod, heat maps from the polymod project, showing the contacts that participants make with other people of other ages in the population. And you can see the assortative mixing that goes on generally. So there's two, there's GB and Poland. They're not the same. They won't be. You can't take a contact matrix from this context and move it and use it in another context. So you have to do these separately for different countries. And when you dig into those contact patterns, you see different patterns in the household. So the panels on the right there, they're from a later study in Germany. And you can see contacts in households, predominantly children and adult type contacts going on, and workplaces and in school. So, and even when you look at different size households, you'll get different contact patterns. So transmissions of pathogens through populations is really dependent upon how populations mix and how populations move. So the left-hand panel there, that's the net journeys into and out of small areas in London. These are small areas, roughly about 8,000 residents, over the course of the day. And the right-hand panels there are starting journeys to two destinations in England. So I lifted these from some tick work that somebody was doing. So I thought they demonstrated that populations are really mobile. You can have a contact with somebody at place X, but that doesn't mean necessarily that if a transmission event has occurred, that won't be reported at where that place is. They will have moved somewhere else. They go back home, become ill eventually after an incubation period. So mixing and movement and contacts really drive the epidemiology of most pathogens. So what happens when we have things like COVID? So this was a shock. I imagine most of you have looked at your time series from your surveillance data and saw something unusual goes on during COVID because there was regulations in place in most countries. We had sort of fairly strict lockdowns in the UK, so we were only allowed out of the house for 1 hour to exercise. And we had various workplaces were closed. You can't close every workplace. There are key industries. You can't close hospitals. You can't completely close schools because of the key workers, children have to be going to school. So it reduces lots of contacts. But it also changes people's health-seeking behaviour. It changes in healthcare provision. changes in biological testing, much more multiplex PCR testing going on post-COVID than it ever did pre-COVID. And all of that makes your time series from your surveillance data quite difficult to interpret. So one of the things we did as part of COVID, this was really work on real-time modeling of the pandemic. So as we were needed to make nowcasts and forecasts for government to base policy and decisions around, we had to understand how contacts were changing. So, and these are the relative reductions in the numbers of contacts for closing different parts of society. So you see, you close the schools, unsurprisingly, you can estimate that will cause around a 57% reduction in contact between school-aged children. You close workplaces, that doesn't really impact schools so much. Children's contact doesn't really change from closing workplaces, but adults do. Social visits, teenagers and the elderly get impacted most in terms of reducing contacts. Closing bars, again, doesn't affect school-aged children, but it affects the elderly more. And you can put those together to understand how contacts were changing dynamically and use that in mathematical models to predict what's happening now and what might happen in the future. This was the impact it had on laboratory reporting. So this is the total number of reports into SGSS, the second generation surveillance system in England. And you can see at that first lockdown in March 2020, there was a massive reduction in the number of reports flowing through that surveillance system. Some of it was due to people not showing up, not going to seek healthcare. Some of it was due to change in epidemiology. But as we come out of lockdown, things change. And you can notice that post-lockdown, so we're actually seeing more reports into SGSS now than we did at the time of the first lockdown. So things have changed. You can sort of look at that and you can say, could I fit a simple model like the flexible Farrington algorithm to that? And it's challenging. You can look at these by specimen type. So positive blood specimens are now higher than they were pre-COVID. They used to run around 5,000 a week. Now they're up somewhere at 7,000. The striking one there is the lower genital tract. So that you can see there that there is it doesn't recover. There's nowhere near, even now, at the levels of reporting of positive specimens from lower genital tract. And that's primarily due to changes in healthcare provision, much more online consultations for sexual health services. So what happened with the flexible Farrington algorithm when we continued to run this throughout COVID every week, every Monday, even though I don't think many of the epidemiologists were really looking at it. They had other things on their mind at that time. But we did run them and provided results. Most times we would see some no outbreaks at all. But eventually we started to see flat signals coming back through the Farrington flexible algorithm. But you can see there was this is just one example of running it post-COVID in 2023. And you can see a whole list of things that flagged. And this is the false positive issue coming back. Originally, when we revised the algorithm, that problem was being caused by the way we downweighted historical observations. This time, it was just the shock. So what can you do? So we sat down, thought about this, about what might work. And it's really like sort of an interrupted time series type of approach. So you select a point or a period where the shocks occurred that you can then essentially ignore if it's a period. And then you say, after the shock, it looks like there's been change in epidemiology or changes in other ways people seek health care, and it's changing the way the surveillance is working. So we allow the temporal trend to change. We have incorporated non-linear trends into this. We only ever used to have a single sort of log linear trend. But we assumed the cyclical patterns would remain the same, and we assumed that the variation, or the extra variation in the data would remain the same pre and post. That sorted out to some degree. So that's running the same week with the revised algorithm. And you can see there that the number of flags is much reduced. It makes the life of the epidemiologist much easier if they haven't got to wade through a big long list of potential things to risk assess as to whether they need action or not. Finally, so this is just some visualizations of some recent results. I think these were not this week's, but last week's results from the surveillance system. I've picked just out four of the several thousand that we do, just as examples. So you can see that there's something going on currently with Salmonella mycosoma that's being flagged. And you can see that the red line there is the expected value from the model. The grey line is the observed data. And they broadly sort of agree up until the recent few weeks. And then you've got adenovirus. That one's showing that there's nothing at the moment, there's nothing unusual going on. We're observing something close to what we'd expect to see, well below the threshold for flagging to the epidemiologists. But you can see that the cyclical patterns did change. They're coming back to, and exactly the same with respiratory syncytial virus there. You can see that we had unusual summer peaks, the way we'd never seen summer peaks before, but again, eventually settling down. And this flexible, or the additional flexibility that we've added to flexible Farrington, we call it F3. Well, I don't know what, it's the third variant of these sorts of methods that we've developed. And it seems to be running okay. We're only running this algorithm now in UKHSA. We used the dual run, and now we've stopped the old flexible Farrington results and provide only these results going forward to the epidemiologists. So that was my last slide, so I'll sit down and take some questions.
Thank you very much, André. That was really good. And before we move into questions from the audience, you know, we have some questions already prepared for this discussion internally. So my first question for you is, how can public health institutions with limited budget and human resources adapt this way of data analysis?
It's an interesting question, because essentially you've all got your surveillance data. So you've all got that. You don't need any more budget to get your surveillance data. You've all got computers, laptops, desktops, whatever they, high-performance clusters, whatever they are. You've got compute facilities. And you've got access to algorithms through the R surveillance package. They may not be the appropriate algorithms, but you've got access to them. So all you really need is scientific curiosity. And I would assume that most people in the audience, some of them might think of themselves as public health clinicians, but most of you will think you're a scientist. So you should have a modicum of inquisitive nature that will... So you can look at these things, opportunities like sort of continuing professional development. Well, could I look at these types of methods in the next 6 months as part of my continuing professional development so I become more familiar with them? I can try them out, play with them, see how they work against your surveillance data. Are they working as you'd expect? Because you're all doing some form of risk assessment every week, or every time you look at your surveillance time series, when you plot them out. Is there anything unusual here? It's sort of one of the fundamental activities in infectious disease epidemiology and surveillance is, are outbreaks occurring? Can I detect them? Can I understand what I need to do? Do I act or don't I act? So you're all doing that. So it's just formalizing this and helping you to understand whether there's more evidence to act or not, really.
And so I was thinking in the interest of time.
Yep.
And because we know that how important it is for making decisions, this signal detection, maybe we can move, Martin, to the questions from the audience.
Thank you very much. Andre, we're just going to start with a very quick one from Rebecca Marshall. She says, just a comment, very interesting to see the lower levels of chlamydia post-pandemic.
Yeah.
We've seen the opposite in Ireland.
It depends. I don't think this is. I don't think this is epidemiology. I don't think it changes in epidemiology.
Right.
There is a paper that was published a year or so back, and it's really how sexual health services are now provided in the UK, and much more online. So if you want your PrEP, you don't need to go to your sexual health clinic. You go online. You say, "Oh, well, I'm running out of my pre-exposure prophylaxis," and it's all sorted out online, basically. So there's less opportunities to take specimens, put them through the lab. So I don't think it's true epidemiology. It's just ascertainment and reduced numbers. If Ireland hasn't done anything, if their sexual health services are similar now to what they were pre-COVID, I would expect, knowing sort of most sexually transmitted infections are increasing.
Okay.
So I think the true epidemiology is that they're increasing, and this is just an artifact due to how services are provided.
Okay, interesting, great. In the post-Brexit era, I've said the word, okay. How much data sharing occurs between UKHSA and the ECDC, such that we can produce real-time maps for the entire Europe, especially since populations at risk move between London and the continent?
I think we're in a place now where we can start beginning these sorts of discussions as to how these sorts of results, we provide these results to the epidemiologists in UKHSA. I think Public Health Wales, don't we still, Chris? Or are you still... We certainly run them for Wales, but whether you can access our systems, that's the problem usually. But there's no reason why we couldn't share them more widely. Why other countries that are running these algorithms can share those results. They could then be collated and made available to professionals. Might be a bit scary to make them available to the public because they still need that epidemiological understanding and before you can really understand truly whether something needs action or whether something is just an artifact. But I don't think there's fewer barriers now, I think, to setting up those sorts of initiatives.
Okay, now there was a third question here, but it's just moved around in Slido, and I can't find out where it's gone. So I'm going to have to... It's moved. People are voting on it, and it keeps on moving.
Downvoting it, probably.
Yeah, no, honestly, democracy at work. It's just awful. So we'll just have to take that a little bit later, but it won't be lost, and we'll come back to it. I just need to find it again. Thank you very much.
Thank you.
Thank you, André. Now I'd like to ask Caterina to address how the latest developments in artificial intelligence can help us to improve healthcare-associated infection prevention and surveillance.
Okay. Thank you very much. First of all, I want to thank my moderators and also the organizers for inviting me here today, and I'm delighted to have the opportunity to present you and to share with you the insights into the application of artificial intelligence in trying to mitigate let's say, and contain the healthcare-associated infections. Okay, it's working. We all know that the complexity of healthcare-associated infection surveillance demands in any case, some innovative ways of trying to address the issue of collecting this data and making this available for IPC methods, of course, implementation, of course. And here you can see some results of an interesting systematic review that we have updated from 2018. These are very recent data, not yet published, but that we decided to update because it was interesting to see how increases the numbers of published papers dealing with introducing artificial intelligence method in surveillance. And here you can see the articles divided by, of course, infection, type of surveillance, and also the methodology applied. Of course, more than one methodology was applied in different studies. That's why you can see the light blue line a little bit longer in some specific issue. And the examples that have been used here in the published papers, you can see it could apply to the entire surveillance of hospital-acquired infection, or to surgical site infection, or to sepsis, or to other examples, as you can see my listed here. And the number have doubled since the 2018 till now. We all know that artificial intelligence is major threat in the consideration of the fact that artificial intelligence could be a black box, no? Algorithm included in this, the deep learning methodology, we all know that we don't know how it works at the end. So that's why there are some methods that could be used for improving and having a better transparency of this kind of models, and we will see. But I want to take the advantage of repeat something has been already said regarding the fact that it's very crucial in epidemiology in general, but in artificial intelligence in particular, to use clean data, complete data, because otherwise our output will be in any case, not real and not reflecting the real situation in our settings. So this is the crucial aspect of artificial intelligence in which we should and we have to use data sets that are complete and of course clean in order to be consistent results and output. The artificial intelligence can use and can be applied for acquired hospital infection surveillance, and in particular for infection and prevention control measures in several aspects. So, of course, the main one is surveillance, but we can also use this for prediction, for predicting the risk of acquiring an hospital-acquired infection in our population, or for prevention strategies, or for outbreak management, but also for detection and sometimes also for treatment of these patients. And these cases have been described widely in the literature, and I took one of the examples that could be very interesting to pick up and to describe this. Especially in the prevention and in the surveillance. In the prevention, we have the method available now in order to address the black box problem. And we know that with the new method of explainable AI, we can identify which are the most important parameters that affect the interest, of course, our model and our algorithm. In this case, this methodology is fundamental for AI application in the prevention field and is particularly crucial. Of course, explainable AI can help a lot in having a clear algorithm, transparent algorithm, and to address all the risk factors that could drive to results and to the output of our, let's call again, black box, but is not anymore that black box. So this could improve, of course, transparency. Here are some examples in prevention for your. This is what we have now available in the literature, and I found this article very interesting because it takes into account the prediction using explainable AI, intelligence framework, using electronic health records available, so demographic data, laboratory data, microbiological tests, all information available at a hospital level. It took all this data together, and they came out using retrospective data, and they came out with the hypothesis and with the special trajectory of identifying their patients as potential cases of hospital-acquired infection, in particular with a special focus on bloodstream infections. And this is, important to identify for us the most at risk population in order to be ready and to apply all necessary infection prevention and control measures to specific population. And this is something very, very useful and very interesting. We hope that we will available in the literature soon as a peer-reviewed article. Another experience we have done in our unit, in our hospital, we are in a tertiary hospital with more than 1,500 beds in Tuscany, in Pisa. It's a university hospital and with a lot of activity in surgery. So we were very interested in trying to increase our surveillance of surgical site infections. And we found very interesting, since our electronic health records were not so consistent because were not so complete, so that the quality of the data was not very convincing for us. So we decided, as you know, probably in the Southern European countries, the clinicians love to take notes instead of including parameters in the electronic health records. So we decided to use another potential source of information that was the hospital discharge letters. In the hospital discharge letters, you can find all the history of the patients during the hospitalization, of course, and also some interesting information regarding the retrospective data of the patients. And we decided to apply a natural language processing method using a specific BERT method, specific for the Italian language. And we came out with of course, with some interesting results that I will present you here. Of course, as you can imagine, since you all know, I'm sure, that the surgical site infection do not rely only for the ECDC guidelines on the inpatients in the hospital. So these data were only for inpatients. As you probably know, the surgical site infection surveillance continuing after the discharge of the patient. So, but we are not still dealing with that part. We will discuss later about that during the discussion. And so, as I told you, we use this natural language processing method that you probably know is an intersection of artificial intelligence and linguistics methods. What we did is to develop a specific NLP method for our setting in order to be able to extract the potential SSI in our hospital setting. And what we found is that we started looking at the data of 2020 and 2021. And from hospital discharge letters, as you can see, we extracted around 64,000 hospital discharge letters. So with the keyword filtering method, we extract, of course, selecting from the literature the words. We extract 27,000 hospital discharge letters that potentially containing surgical site infections or words related to surgical site infections. Then, with the help of 40 medical residents in public health that I want to acknowledge here, of course, with their work, we divided these groups and we asked these medical residents in public health to label these 27,000 hospital discharge letters using a specific, we built a specific tool in order to make them more comfortable in doing all the annotation, of course. And then we came out with a total number of approximately 600 SSI identified and method, because these manual labeled hospital discharge letters were then were put in the algorithm to create, of course, into the BERT algorithm created for the national language processing, and we came out with approximately 600 SSI from our dataset, complete dataset of 2 years. At the end, we decided to validate this information, and the case were confirmed with full medical records. And as you can see, we came across a validation of 350 SSI in our setting. Of course, as an epidemiologist, I have to highlight for you the fact that of course, this data set is a little bit unbalanced, okay? Because you have a lot of negative cases and very few positive cases, but with a very complete information about and very complete data set that can be used, of course, ideally for enriching the algorithm and enriching the method. What we will do, our next steps, of course, we will try to include in this method also structured data. So structured data coming from electronic health records or other surveillance systems that we have in place in our hospital. And of course, this will lead also to try to increase our capacity in the prediction outcome. For instance, our final goal is to try to came out with a method able to identify SSI, but to identify also patient at risk to develop an SSI in order to apply all the infection and prevention and control measures to be sure that that patient will not go through an infection in case of a surgery. And another important aspect that we are looking into is the privacy and ethics issue that you can imagine is very important, especially in Italy, where we have a specific law for privacy, and it's even much complicated than the normal GDPR. We have a specific clearance for our ethical committee to conduct all this study, of course, but we know that the only way to enlarge our study and to try to identify the way to standardize this system is to use probably federated learning that made us available to share only the algorithm and not the data. And that's something which we are working at our university, where we have experts in this field, and we hope to be able to have a pilot soon with another hospital in Tuscany that is available for doing this, that has very good surveillance data. So let me acknowledge all the persons involved in this process. These are only few, let me say, because of course the human resources here are very crucial and important in order to identify and submit the goal. And I hope to have covered all the issue and all the aspects, and let's hope that this will help us in having better infection and prevention control measures for our patients. Thank you very much.
Thank you very much, Caterina. You have already briefly addressed resources and capacities, capabilities. Can you please tell us again how public health institutes across Europe with limited resources in terms of budgets or human resources, can introduce the use of these AI tools?
No, I think, as I said, that this, as you can imagine, if you are involved, the hospital-acquired infection surveillance is very demanding. You need human resources, you need people trained for doing that. So the idea of having a system using, everywhere data are available, even in low-resource countries, you can have at the hospital level data available, so you can use the data to train the system and to specifically address this issue in the setting, in the specific setting you're working in. And that's why this method could be very helpful. The only thing that I want to say, and that is, in my opinion, crucial for this, we cannot accept the black box as it is. We should be part of the process. We should be engaged in the process because we have the knowledge and all the knowledge to make the algorithm and the method and the model more precise and more adapted to our situation and to our setting. And this is crucial. So I think that this could be very helpful for low resource settings. And as you can imagine, we adapt the system to hospital discharge letters because we don't have electronic health records of higher quality in order to address this issue using the specific data. So you can find a way. Of course, you need people, experts in the field, and you need a multidisciplinary group in which you can work and develop this kind of methods.
Would you tell us what sort of expertise you would like to see in these multidisciplinary
Of course,
groups?
you need clinicians, you need epidemiologists, you need infection prevention and control experts, and you need, but you need also those that are the new brand statistician, the data scientist, also, and experts in artificial intelligence methods. And also there, artificial intelligence is a big whole. Inside that, you can have machine learning, deep learning, NLP, so natural language processing methods. So you should have experts depending on what you have to do. So it's important that you have a clear idea of what you are doing and what are the best methods to address this, that issue and that aspect.
And can this lead us to better decision making?
Of course, that's the ultimate goal. So, and of course, in terms of better decision makings, I have to say that the opportunity to having also this kind of methods in place make your surveillance also more timely than the normal manual surveillance. And this can lead, of course, to implement better prevention and control strategies and to manage better even outbreaks or complicated situations like the introduction of a new agent in the hospital that can be also deadly, like Candida auris, for example, or other kind of multi-drug resistant microorganisms that, having in place such methods coupled with all the stuff, with all the things that we have listened today, I think could be the right way in which we can identify and address the infection control at the hospital level, but not only. This is an example of the hospital level, on the hospital level, but we can also enlarge this then to the nation level, hopefully.
Thank you so much. I'm quite sure that we have questions from the audience, right, Martin?
We do, thank you. First question, Caterina, you emphasized that it's crucial to use clean and complete data. But in real-world epidemiology, how realistic is it to have such data?
No, it's not realistic at all, I know. But important things is that if we are dealing with this kind of model, to, let's say, to increase the capability of the model to have a reliable output, the important part is that for that reason, you use clean and validated data. So the important aspect is to use clean and validated data and a clean data set and a validated data set for the meaning of the producing the algorithm. Then once the algorithm is ready, you can use with every type of data available to address, using your data, of course, to address the issue that you are looking for. This, for the SSI, was an example, but you can use for CLABSI, catheter-associated bloodstream infection, or catheter-associated urine tract infection. So it's important to train the model with good data. Then it can be used also with not so good data, let's say.
Well, building on that question, or building on that issue, pre-existing biases in the training data, based on sex, race, for example, can get propagated through AI models. So how do you make sure your model avoids these pitfalls and remains fair and equitable without enshrining pre-existing discrimination?
Now, with explainable AI, as I mentioned, this kind of issue could be adjusted, let's say. And so this kind of confounders that we are habits to talk about can be very well addressed, because you can see all the potential confounders within your algorithm. So you can describe what's going on in the algorithm. Different is when you use deep learning. If you use deep learning, that's impossible. So you can't really rely on what you are doing if data are not consistent of high data quality and can address all the issues that you have listed, like ethnicity and everything.
Final question. Could you elaborate what is revealed with explainable AI?
Yeah.
Is it the importance of individual features in your input, importance of certain layers of the network, or their connections?
Okay, it's a mix. It's a mix. At the end, we can, with explainable AI, we can surely look into all these aspects, but in particular with the individual features, especially at the hospital level, it could be very, very helpful in using explainable AI. This is something I think is quite recent, but I think it's crucial to be applied, especially when you deal with the AI in your field, let's say. I want to say.
Wonderful, thank you very much. And we just have time to come back to the question of Michael. I apologized, I lost it. This time I took no chances. I've actually written the question down. So, Andre, back to you. With the shock, is it just a drop in the baseline, but similar trends and seasonality post-shock? If that's the case, do you not just reset your model and ignore the pre-shock data to calculate your expected values, or are the patterns different altogether?
You can reset. You can ignore everything pre-shock and during shock if you want to. That usually limits you for a long time because you need a reasonable amount of data. You can probably get away with 3 years worth of data. But as we saw with certain examples there, RSV, cyclical patterns, the epidemiology was completely disrupted, even post-shock for a while. Mumps, we haven't seen mumps resurgence yet in the UK. I'm just waiting for it at some point in time. We know it will happen. But the surveillance data at the minute is flat as a pancake. We're not seeing mumps. We've seen measles. We've seen pertussis. We've seen hepatitis of unknown etiology, which was AAV2. Eventually got to the bottom of that. So you can do that, but you're always going to have to make some assumptions. As I say, there's different ways of doing it, and if you had enough time, you could actually try different ways and see what the performance was like, and plump for one that was performing as you wished it to, or as close as to you wished it to was possible. But it's playing games really with the data and to trying to understand how you modify things.
Very good. Yeah. Thank you very much. Please, back to you.
Thank you, Marty. Thank you, André. Thank you. And so before we finish, we have one final question, but this time the question is for you. So if you don't mind, please go do the Slido thingy. And find the question for this session. The question you will see there is, how confident are you that the surveillance techniques we have described here can be implemented in a similar way across Europe in the next 5 years? And you can select between not confident, somewhat confident, and confident.
It depends.
Confidence, 90%.
We need an interval.
You made your bets about the outcome?
We are doing.
Alicia, can we see the results, maybe? Yeah.
Still.
You can still vote.
Yeah. 300 answers so far.
So far,
As it appears.
somewhat confident.
This number of confidence is increasing.
Has the highest percentage.
Yeah.
Not confident, confident. The two opposite poles have a similar percentage. Somewhat confident is, how they say, nice outcome. There's some hope. Caterina, what would be your final message to encourage people to be more optimistic?
You think we can increase the optimism? I have to say that when I started my work as an epidemiologist of infectious disease in the National Institute of Health, we started and it was very difficult to implement some specific surveillance methods, surveillance systems. Now we have the data, we have the methods, so I will feel more confident than not confident on the implementation of such strategy. We should work and we should look into, and we should work more in order to be able to implement all the systems that we have heard in all the possible settings where we have resources, human and funding.
Human and funding.
Yeah.
André, what would be your final message?
I think it's really around, well, from the sort of the outbreak or the statistical algorithms for outbreak detection, it's really around trying to become familiar with them and what they're doing. And when you do become familiar with them, you can modify them. You get other questions, what goes on at a local area as we come out of lockdown. So you can modify these algorithms, and it's exactly what we did. So it's just trying to have that time to familiarize yourself with what's out there, what's available. Are they fit for purpose for you? Do they work for you? If they don't work for you, how might you be able to modify them so they do? So it's just basically that learning process, that experiential process that you need to go through. There's no shortcuts to it, really.
It's a learning process.
Yeah.
And Lieke, your final remarks.
Yeah, I think for sequencing, it's exactly the same. Like all the tools are out there and all the capabilities are out there. A lot of the tools nowadays, especially for the data analysis, are freely available and you can search them, find everything online. Yeah. So it is becoming a lot more easy nowadays to just start doing it also with decreasing cost for equipment. It's easier to just start playing around with it and then build from there, I think.
Thank you. Thank you, Lieke. Thank you, André. Thank you, Caterina. This was this. You want to clap? Not yet. So we hope you have enjoyed the session. It was our intention to bring together, talking about surveillance, these main two pillars that are microbiological data and epidemiological data, and put together now in this picture this artificial intelligence that is not that's coming, it's somehow already there. And our intention was to focus on recent examples, real situation that's happening in the state of the art nowadays in our field, and highlighting the importance of the data sharing, the harmonization of methods to be able to determine whether this cluster is an outbreak, to use the surveillance algorithms that can be modified, not that scary, and the understanding of this black box, hoping that with understanding the data that we feed in, the black box would be less obscure in our world. So at the end, the main message is that surveillance is information for action. You may have heard this before, and we can use these tools to help us using this information. Yep.